Hi, my name is Rowland-Onyekachi, and my research is on targeting MCT1 and 4 proteins in MDA and B231 cells to reverse lactate-mediated immunosuppression. Now, cancer cells are known to prefer glycolysis as the mode of metabolism because it generates ATP faster, but also produces a lot of lactate as byproduct. Lactate can be used as fuel by these cancer cells, but can also be harmful to them, especially when accumulated over time. So to prevent that, these cancer cells use proteins called monocalculate transporters, also known as MCTs. Now, MCTs can either transport lactate in or out of the cells, and cancer cells can use lactate as fuel, or they could send it out into the tumor microenvironment to prevent immune cells from detecting them. So we thought of, what if I try to trap this lactate within the cancer cells so it kills them? Now, quercetin is a compound that is found in a lot of fruits, natural resources, and it has been shown to have anti-cancer effects on MDA and B231 cells. So I decided to treat my cells with quercetin and to study the effects on MCT1 and 4 activity. So the first test I did was extraction. I used ultrasonic extraction to extract quercetin from red onion skin. And then I used the extract to treat my cells for 24 and 48 hours. And the cell viability assay showed that 48 and 24 hours at 41 micromolar concentration had significant decrease. And at 4.1 micromolar concentration, there was increase in the viability, though not significant. This increase could be because the concentration of quercetin is low, and so therefore it was acting as an antioxidant and improving the metabolic activity of the cell. Now, the second assay I did was the migration or the scratch assay. So I treated my cells with 41 micromolar of my onion extract for 24 to 140 hours. And I had a significant decrease in migration over time. And this confirms with my hypothesis that quercetin would inhibit the migration of the cancer cells since they're dying. Then the DAPI assay was the third assay I ran, and it also showed that there was increase in apoptotic cells compared to the control. Now for my PCR, I checked the expression of SLC16A1 and SLC16A3. SLC16A1 is known to control MCT1 and SLC16A3 controls MCT4. So in SLC16A1, I found a decrease in expression, though it wasn't significant. But in SLC16A3, there was a significant increase in expression. Now, this is contradictory to my hypothesis because quercetin is supposed to downregulate the activity of this gene, thereby trapping lactate within the cells. But in this case, it could be said that quercetin was inhibiting SLC16A1, but then since cancer cells are very good at adapting to adjust to cytotoxic conditions, they decided to upregulate SLC16A3, which is known to export lactate in the remaining viable cells. So more lactate was being exported at 48 hours from those viable cells. So in conclusion, I could say that quercetin did have cytotoxic effects on MD and B231 cells, and it did downregulate the MCT1 activity. So further research can explore more natural products that have quercetin in them and can also see the gene expression of MCT1 and 4 over longer treatment conditions. And yeah, thank you for listening.